and I say thank you to everyone for your patience. Ronja, can you please start the YouTube live stream? And we kick it off with this public lecture. I'm very happy that you've been so patient, and we can start. I have one remark that I want to make before I hand over to Manfred Weiss. We will have opportunity for Q&As, and since we have people that are not in this platform and are joining us remotely, there is a link. So if you just write that down, bit.ly BESSY User Meeting, and you can enter that, and there you can post your Q&As. It's also in the description of the YouTube link. So if you go to read more in the description, you see the link there. And with that, I'll leave it. Everyone's invited to use this link. I'll also post it in the chat here, and we're looking forward to many Q&As. And with that, I hand over to Manfred Weiss and Professor Hilgenfeld. If you would double click again on your slides, then you're good.
Okay, good evening, everybody. Welcome to the public lecture of this year's HZB User Meeting. Yesterday, I've learned that the most Googled word in 2020 is coronavirus. And so this has, of course, affected our work as much as the life of everybody else. And this is why I think it is most appropriate to have this also as a topic of the public lecture of today's user meeting. Now, I'm very grateful that Rolf Hilgenfeld has accepted our invitation to give this public lecture. So before we start with this lecture, I would like to give a short overview of his biography. Rolf studied chemistry in Göttingen and Freiburg, and then did his PhD with Wolfram Sanger at the Free University of Berlin. Then moved to Frankfurt to build up the protein crystallography at the company, which at that time was called Höchst. Then moved to Jena to a newly founded institute in former East Germany at the Institute of Molecular Biotechnology. Since 2003, he's professor at the University of Lübeck, and so since then he was also the director of the Institute of Biochemistry of the University of Lübeck. He's retired since end of March 2020, but University of Lübeck were very lucky to be able to hire him as a senior professor and enable him to continue his very timely research. Now, a few more things. He became honorary doctor of the University of South Bohemia that's Budweis in the Czech Republic for his work on SARS-1. That was in 2009. From 2010 to 2012, he was a visiting professor at the Chinese Academy of Sciences at the Shanghai Institute of Materia Medica. And in 2015, he received the Gehong Medal of the Wuhan Institute of Virology. This just shows that his affiliation to Wuhan and SARS and coronaviruses in particular dates back much longer than the actual SARS-CoV-2 outbreak. I know Rolf personally for a long time. He's been working in structural virology for the better part of 20 years, I guess. And today we're going to learn that it actually pays off to fund research in areas which may not be so popular at the time, but which have the potential to gain an enormous popularity, as we have seen with the SARS-CoV-2 outbreak this year. And this experience, of course, puts him in a prime position to become very successful in his research, in particular with SARS-CoV-2 now. So Rolf, the stage is yours now. And I'm very much looking forward to your talk on structural biology of coronavirus, in particular SARS-CoV-2.
Thank you very much, Manfred. Thank you for the invitation. It's a pleasure to be here this evening. So sorry for the technical problems. Can I have my slides? You make that full screen. Well, let me get started while the technician is working on this. So, as Manfred said, today we talk about structure-based drug design against the main protease, the key enzyme of the SARS coronavirus 2. Before I get started, I'd like to acknowledge my funding sources. We are part of two major European projects. One is called SCORE, the other one CARE. These acronyms obviously stand for something like rapid response to coronavirus. I never can remember the meaning of the acronyms. But especially the CARE project is a huge project with 37 partners. Total funding is 77 million euros. And almost all of Big Pharma is participating in this. So there's big hopes that finally pharmaceutical industry gets interested in coronaviruses and is investing heavily, and hopefully something will come out of it in the next 5 years. We are also funded by the BMBF through isolated projects and through the German Center of Infection Research, DESIF, which is a virtual institute of which we have been a founding member since 2012. And we have support from the University of Lübeck. Now, everyone these days is talking about the vaccination. And obviously, to have a vaccine would be very important to stop the current pandemic. But even though all the news are full of hopes and vaccination programs will start as soon as in a few weeks from now, we don't know much about these vaccines. We don't know whether they will protect us only from disease or also from infection. So it could be that even though we are vaccinated, we will be able to get infected, but remain largely symptomless and continue to spread the virus. That is still a big unknown. We also don't know whether we will be protected for 3 months or 6 months or 12 months. Nobody had a chance to explore this. And if it's only for 3 months, the vaccines will not be sufficient to stop the pandemic. Even 6 months could be too short. Further, not everyone will be ready to get vaccinated. A recent survey by the MDR television showed that only 39% of the people questioned will be ready to get vaccinated. But to stop a pandemic, we need a vaccination rate of 70% of the population. That will be very difficult to achieve. So, on top of the vaccines, we need antiviral drugs, which can heal those already infected and prevent further spreading of the disease. And antiviral drugs can also help to protect from infection. If, for instance, you have someone infected in a family, then you give the antiviral drug in a protective manner to other family members, and this way prevent the spreading of the virus. Now, if we look at the time course of the pathogenicity of SARS coronavirus 2, we see, and unfortunately I don't have a pointer, so I have to give this talk without a pointer. The software doesn't allow this, but I try to guide you through my slides. We see that the peak of viremia, the highest virus levels, occurs after about 5 days after infection. But symptoms typically only appear 4 days after infection. So we need to act quickly if we want to develop and administer direct-acting antivirals. Because after about 10 days to 12 days, the level of virus already declines in the patient. There is, in most patients, a persistent level of viremia for a longer time, as you can see from the red line to the right of the graph. But now the virus levels are lower and other phases of disease take over. So in many patients, we have a cytokine storm phenomenon, large production of cytokines like interleukin-6, which leads to severe inflammation of the lungs and lung damage. And people actually, if they die from phenomena such as this, cytokine storm and hyperinflammation, rather than from the virus itself. So it would be important to have drugs lowering the inflammatory response, lowering the level of interleukin-6 and TGF-alpha and beta. Also, a phenomenon which has first time really been observed with SARS coronavirus 2 is blood coagulation, massive coagulation of the blood, and people can die from that as well. So you need blood thinners in addition to any antiviral that you provide. So combination of therapies may be the best solution to combat the infection. So it makes sense to give antivirals for a long time because some patients shed the virus for a long time, but to achieve the best effects, we have to act quickly and give the antivirals soon after infection. That means we need to have good diagnosis. So the rapid tests, which are under development now and actually are getting available now, are a big hope for their combination with direct-acting antiviral drugs. Now the next slide. This shows the targets the genome of SARS coronavirus 2 offers. The RNA genome of this virus is the largest RNA genome known. I mean, in general, of coronaviruses, they have a genome of 30 kilobases. And there are some key enzymes encoded, but first, the RNA is translated in the infected host cell into proteins. From the 5' 2/3 of the genome, two large polyproteins are made. These are indicated in red at the top of the slide. These are called PP1A and PP1AB. These contain thousands of amino acids. And these two large polyproteins are processed by two viral proteases into the building blocks of the replication transcription complex. So that is why the proteases are essential for replication, because if the polyproteins are not processed into their building blocks, then replication cannot occur. The two proteases are the papain-like protease, indicated in blue here, which itself is a part of the huge polyprotein, the oligoprotein, NSP3. NSP stands for non-structural protein, because these proteins are not part of the virus particle. They are only expressed in the infected cell. And the so-called main protease, also called 3C-like protease, which is indicated in red. And our target today is the main protease, the red enzyme, which is also called non-structural protein 5. In addition to the two polyproteins, there are the structural proteins, which are encoded in the 3' terminal third of the genome, in particular the famous spike protein, which interacts with the cellular receptor, angiotensin converting enzyme 2. Then the nucleocapsid protein, N, which encapsulates the viral RNA, the membrane protein, M, and the envelope protein. On top of that, there's a couple of accessory proteins, which are fairly complicated, poorly understood in functional terms, and also in structural terms, which interact with host proteins to modulate the innate immune response of the host. So now let's talk about the main protease. Structural biology of coronaviruses did not start in January or February this year. It started 20 years ago when at that time in Jena, my group determined the structure of the first 2 coronavirus proteins, the main proteases of the porcine coronavirus, transmissible gastroenteritis virus, and the human coronavirus 229E. And I remember at that time the scientific advisory board of my institute in Jena asked me to stop the research on these unimportant coronaviruses. But I did not follow the advice because I knew that these viruses only caused at that time mild flu-like symptoms in humans, but they killed the young piglets to an extent of 100%. That is true for transmissible gastroenteritis virus. So we tried to understand the reason for that deadly pathogenicity in animals. And we were already prepared. I looked back into my old DFG proposal of 2002 that one day these viruses might jump over to the human population and cause huge damage. And that happened 1 year later with the SARS outbreak in 2003. So we were already in a position to determine the structure of the SARS main protease as well in collaboration with Zia Rao at Tsinghua University in Beijing. And so the enzyme that we found was a dimeric enzyme, and we made sure that the dimer actually is the entity which is required for enzymatic activity. And you see in this picture a green monomer and a purple monomer. So each of the monomers has 3 domains. From the N-terminus, we have 2 beta barrel domains, which harbor the active site consisting of a cysteine residue and a histidine residue in a cleft between them. And then we have a C-terminal alpha helical domain, which is involved in the dimerization. This slide also shows the cleavage specificity of the protease. These proteases predominantly cleave after glutamine residues. And we call this glutamine residue P1. And the corresponding binding site on the enzyme is called S1. And then also P2 has to be hydrophobic. So you mainly find leucine, phenylalanine, or methionine in this position. P3 doesn't play much of a role, and P4 has to be small. Also P1 prime on the other side of the cleavage site has to be small, either glycine or alanine or serine. So we modeled our first peptidomimetic inhibitors to follow this specificity pattern. And the good thing is that there's no known human protease which has the same specificity pattern. So we have a good chance to come up with drugs that have little side effects on humans. Now, this is one of the first inhibitors. Actually, we did that, synthesized that. Shaila George did it. That's why it's called SG compounds in 2013. And you see, if I guide you through from the right of the formula, that this is an alpha-beta unsaturated ester. So you have the ester group to the right, and on the left side of the carbonyl, you see a double bond. And this is the point of maximum electrophilicity of this inhibitor. So the active site cysteine of the enzyme will attack the beta carbon 2 positions from the carbonyl group. It will not attack the carbonyl group itself. These compounds are called Michael acceptors, and they are irreversible inhibitors. The position left to the double bond, you see this cyclic lactam structure. And if you can imagine, remove the cycle in that side chain, then you see the glutamine side chain. So we use this as a mimic for glutamine. The ring-like lactam makes the structure more rigid, and we lose less entropy upon binding to the enzyme. This increases the affinity, according to our measurements, by a factor of 10. And the position left to this, you see this benzyl group derived from phenylalanine. And this is a large hydrophobic group. And actually, it is non-ideal for coronaviruses. It's big to large, as we'll show you in a moment. But we used this because we wanted this compound to inhibit also the related 3C protease of enteroviruses, like human rhinovirus or Coxsackie virus and others, because there's much more disease, at least until the end of last year, caused by enteroviruses than by coronaviruses. So this way we hope to get the pharmaceutical industry interested, because enteroviruses were a market even before last year, but coronaviruses were not. So we made broad spectrum inhibitors using these Michael acceptors. And this slide summarizes the mechanism of the Michael acceptors. So, and the alpha ketoamides, which we are working on now. So on top, you see the reaction underwent by Michael acceptors and the covalent bond formation between the active site cysteine and the beta carbon of the alpha-beta unsaturated ester. In the lower slide, you see the alpha ketoamides, which we are working on now. And to guide you through this formula, again, looking from the right, you see an amide, and you see two carbonyl groups next to one another. One, the left one, belongs to the amide group, and the one to the right is the alpha keto group. That's why these are called alpha ketoamides. And the alpha carbon of that second carbonyl group is a point of maximum electrophilicity. So the active site cysteine will attack here and form a thiohemiketal. And this reaction is reversible. So in contrast to the Michael acceptors, which are irreversible, the alpha ketoamides are reversible. And they are milder electrophiles. And we wanted to use milder electrophiles to reduce the side effects on host cell cysteine proteases and also serine proteases. So alpha ketoamide is the warhead, as we call this, the warhead of choice for our program at the moment. Now, because we had worked on coronavirus main proteases for over 20 years, we were very fast when the new virus came up. And from December 31, when we heard the news from Wuhan, we were waiting for the genetic information. And that was published by Chinese colleagues on January 11 on the internet. So we looked for the sequence of the main protease of the new coronavirus. And once we had it, we ordered the gene from a commercial company, got it delivered. That actually took the longest time in that period, took almost 10 days. Then we cloned it into E. coli, expressed the protein, purified it, crystallized it. We had crystals by end of January, within 2.5 weeks of the publication of the sequence. And then Manfred gratifyingly gave us beam time on one of his beam lines at BESSY. And we traveled to Berlin with the crystals. That was done by Linlin Zhang, my co-worker. And she collected data with Manfred and his colleagues. And she solved the structure the same day and refined it in the days to follow. We also co-crystallized the enzyme with an inhibitor, as you shall see in a moment. Now, what are the differences to the SARS coronavirus 1 main protease? Very few. The sequence identity is 96%. And I would say there's only one important difference, and that is indicated by the 2 black spheres in the right image. And this is a mutation in the C-terminal dimerization domain. We have a threonine here in the SARS-1 main protease, but we have an alanine in the SARS-2 main protease. That looks like a small difference, but the threonine in the SARS-1 makes a hydrogen bond between the two monomers. This is a symmetry-related site, so the twofold axis of symmetry runs between these two residues. The alanine in SARS-2 cannot do this, so there is a little bit more space. And that has the consequence that the two domains 3 can approach each other more closely. And somehow, and this has been predicted by molecular dynamics simulation by Dr. Song in Hong Kong years ago, this is transmitted to the active site, and we get an increase in catalytic activity of a factor of about 3. Excuse me. Now, we had this inhibitor, this alpha ketoamide, and we, our prime alpha ketoamide design was a compound which we called DZL901. DZL stands for Daisong Lin, who synthesized it in my lab. And we co-crystallized this compound with the SARS-2 main protease and found good electron density and good binding. So in the left image, you see the formation of the covalent bond between the active site cysteine 145 and the alpha keto group. Then left to it, you see the P1 residue, that lactam glutamine mimic binding to the S1 pocket. It neatly fits to that pocket. And then to the left of that, you see the benzyl group bound to the S2 pocket, that hydrophobic pocket. Now, this is not only a lock and key process, this binding process, it's actually induced fit. Because in particular in the S2 pocket, the enzyme has a certain degree of plasticity. And if we have a smaller P2 residue, like in the right slide, we have a cyclopropyl instead of the phenyl ring, so much smaller. We see that the pocket shrinks. And this is achieved by a movement of that residue, which is indicated as Q189, standing for glutamine 189. And glutamine 189 is very flexible. And it can either make a hydrogen bond to the inhibitor, as in the right slide, or if it is pushed back by a P2 residue, which is as large as the benzyl group, then it does not make a hydrogen bond. So simple docking is actually not working very well here because of the plasticity of the enzyme. Now, to find out more about the structure-activity relationship of the P2 residues, we made a series of inhibitors which have different ring sizes in the P2 position. And unfortunately, I cannot point this out, but please have a look to the center of the formula, and you see from top that we have either a benzyl group, that means phenyl group plus CH2 in this position, or we have an isobutyl group, or we have cyclopropyl, or we have cyclobutyl or cyclopentyl. So we tried to find the best fit, and it turned out that the best fit for the coronaviruses was a small cyclopropyl. So we developed our inhibitors further. Up to mid of last year, our lead compound was 11R, which is an open chain inhibitor and alpha ketoamide, and it had the cyclohexyl methyl group, the 6 ring, in P2, because it was also made to inhibit the enterovirus 3C protease at the same time. It had a relatively short half-life, less than 1 hour in blood plasma, and the pharmacokinetics was okay, but not superb. So we wanted to increase the half-life. And the first thing to do if you have this problem is that you remove some of the peptidic character of the compound. So we tried to hide the amide bond between the P3 protecting group, that's the group to the left of the formula, and the P2 residue, the cyclohexyl methyl group. And to offer less attacking points for host cell proteases and increase the half-life of the compound. So we were hiding the amide bond in a cyclic pyridone structure. You see this pyridone, the cyclic structure with a carbonyl pointing down to the left of the formula. So this amide is not easily accessed by host cell proteases. And this we call a second generation alpha ketoamide. And this was compound 13A. And then when we had co-crystallized 13A with the SARS main protease, we saw that the cyclohexyl methyl is a bit too large for the S2 pocket. So we replaced it by the cyclopropyl. This created compound 13B, which now has a half-life of 1.8 hours in blood plasma. It has good bioavailability as expressed by the AUC, the area under the curve, and it's not toxic to mice. So this shows the crystal structure, which we also collected data for at Manfred's beamline at BESSY on February 1 this year. And you see the pyridone ring-like structure indicated in black in the electron density for compound 13B. So everything fits nicely. The IC50 for this compound was 0.67 micromolar, 670 nanomolar. All right, and then we of course had to test the compound in virus-infected cell culture. This was performed by Stefan Becker's group in Marburg, and he used CALO3 human lung cells, and he found an EC50 of about 4 micromolar. So not superb, but it was efficient. Actually, quite interesting is that we needed the C-terminal protecting group of 13B to enter the cells. That Bock group is hydrophobic. When we removed it to create compound 14B with a free amino terminus, there was almost no antiviral activity. So a small chemical detail can be very important for entering the cell. We then gave the compound to Katharina Rox at the Helmholtz-Center for Infection Research in Braunschweig, and she did pharmacokinetics in mice. And she found that first, the compound is not toxic. Second, there was a nice relationship, linear relationship between dose and activity. She found it possible to infect the mice by subcutaneous injection 3 times a day, every 8 hours. That achieved useful levels of the drug at all times higher than the IC50 against the enzyme over the day. And in particular, there was a tropism for the epithelial lining fluid, which surrounds the lungs. And this is where the virus mostly assembles. So this is where you want to have an antiviral compound. She also did applications through inhalation, and that also worked nicely. So 2 inhalations per day were necessary to achieve good levels. It would be useful to increase the half-life of the compound further. 3 injections subcutaneously per day is a little bit too much. You want to reduce it to 2 injections, and we are working on that with the next generation of alpha ketoamides. So the pharmacokinetics of 13B overall are favorable for further development. Now, there are 3 stereocenters in compound 13B. These are indicated by stars here, by the little asterisk. You see, one is inside the lactam ring, The second one is connected with the C alpha of the P1 residue. And the third one is connected with the C alpha of the P2 residue, the cyclopropyl methyl group. What about diastereomeric purity? According to the synthesis scheme, the reaction which we use to create the lactam should be stereospecific. So we expect at least 95% S configuration for both these stereocenters in P1. But the P2 turned out to be a problem. And while we thought we have pure diastereomer here, this was not the case. And finally, in collaboration with the British company Tocris, we managed to separate the diastereomers at this position P2, and we first were able to crystallize the R diastereomer in this position and separate it from the S diastereomer. And this is a crystal structure with a pure S diastereomer, which is supposed to be the active diastereomer. And after the separation, we found that it had about IC50, about 50% of the original one, 0.33 micromolar. So it was double the activity of the original racemic compound. And you see again the pyridone indicated in black here. Obviously, the structure is almost identical to the one we had crystallized before, because the enzyme had selected the active SSS compound in the crystallization complex. What about the inactive, supposedly inactive R diastereomer? Now, that structure is shown on the right slide. The IC50 for that compound, the RSS diastereomer, was 15 micromolar. That, in our eyes, is inactive, even though people publish papers when they have an IC50 of 20 or higher micromolar. Excuse me. On the right image, you see the crystal structure with the inactive diastereomer, and that is very interesting. Let me guide you through the compound from the left side, from its end terminus, so to speak. At first, you have the protecting group in the P3 position, then you have the pyridone ring, and you see the P2 cyclopropyl methyl. That one is the one with the wrong stereochemistry at C alpha, cannot insert into the S2 pocket. And therefore, it points out of that pocket. It points out of the screen here towards you. And that's why the activity is low. The P1 lactam has the correct stereochemistry, and it binds properly to the S1 pocket. The alpha keto group also changes stereochemistry from S to R. It is obviously, after the reaction, also a chiral center. And then the inhibitor folds back into the S2 pocket. So the S1 prime benzyl group, which is supposed to bind to S1 prime, is now binding to the S2 pocket. And even though the compound is inactive, it tells us a lot. It tells us that first priority is to fill the S2 pocket with a good hydrophobic binder. And second, the benzyl group is probably too large for that small S1 prime pocket on the other side of the active site cysteine. So the next round of inhibitors, which we are making right now, we have smaller groups there, or even no group. Also, this semicircular arrangement of the compound might suggest that it may be interesting to make cyclic compounds, and by doing this right now, to increase their stability in blood plasma. So we can only emphasize the importance of pure diastereomeric compounds. And this is a collaboration with Tocris, which is a subsidiary of Biotechna, and the pure diastereomers will both be available from Tocris from next year. Now, other people have not been sitting idle since we published our paper in Science in March this year. And the most advanced compound is probably the Pfizer inhibitor, which is a simple ketone. You see the ketone group to the right, also a mild electrophile. This had been developed by the company during the SARS-1 outbreak, but then was halted because there was no further interest in coronaviruses. You see the compound is very peptidic. They didn't make any effort to hide the amide bonds, nothing like a pyridone here or similar. So as a consequence of that, the oral bioavailability is less than 2%, and the compound has to be given intravenously, which means the people have to be in hospital for the treatment. But of course, this will be very useful to treat the severe cases. And it's in clinical trials right now, but we haven't heard much about the results yet. You see, they also use the cyclic lactam in the P1 group as a glutamine mimic. So that is a common principle now. And this shows the crystal structure of the Pfizer compound with the target enzyme. Now, alternative approaches. Obviously, the development of these drug-like compounds that I described so far, these leads, will take years. We are entering animal experiments in the hamster and mouse model for COVID-19 right now with our purified diastereomers. So we are certainly years from market introduction, probably at least 5 years. So we need something to treat the current patients, which can be developed faster. And here the keyword is drug repurposing. Can we use the thousands of drugs that we have at our disposal right now to treat coronavirus disease? One approach is virtual screening. What you do is you take your crystal structure and use a chemical library of thousands and hundreds of thousands of compounds to screen it for those compounds which theoretically can bind to the active site or other functional sites of your target enzyme. And we did that in collaboration with Scott Egan's group at University in California. And we screened the zinc library, which contains 230 million compounds. And we filtered out the drug-like and commercially available compounds among these. These were almost 10 million compounds. And we used the iDock program to dock these compounds into the active site of the SARS-CoV-2 main protease. And we found a number of useful hits, but only about a few dozen hits. And in the inhibitory tests with the recombinant enzyme, only 2 showed moderate activity. And you see one of the formulas shown down there. And you see that the compound has a lot of nitrogens, which can act as hydrogen bonding acceptors or donors. So that is how, according to the model, it binds to the active site. But overall, the result of that huge computational activity was disappointing. And that is what other people tell me who do virtual screening. They come up with a lot of compounds. Many of them proposed collaboration to me. We even bought some of these compounds for a lot of money. And we found, yes, the compounds bind to the enzyme, but they cannot compete with the formation of the covalent bond between the active site cysteine and the polyprotein fragments, the formation of the acyl enzyme. That creates a lot of binding energy, where the non-covalent binders, which you typically find in this virtual screening actions, cannot compete with. So that approach was overall disappointing, although I can agree that if you have a library of specialized binders, then you may be more successful. Another approach to drug repurposing was the big activity started by the BESSY colleagues. And they used the Fraunhofer repurposing library to screen the binding of more than 5,900 drug-like compounds, which were either FDA approved or are in clinical trials or have been in clinical trials to test for binding to the main protease. And their approach was co-crystallization. They took our Mpro gene, expressed the protein, purified it to a very high degree, crystallized it, and co-crystallized it with the candidate drugs. And they found 37 of these 5,900 plus drugs bound to the main protease in the crystal. And of these, 6 compounds showed antiviral activity in virus-infected Vero cells. But for most of them, the EC50 was above 20 micromolar. So in my eyes, inactive. 2 of them had higher activity, and they are listed here. One is a known inhibitor of the main protease, calpeptine. It inhibits many cysteine proteases with low specificity, also calpain, that's where the name is derived from. And the other one is an anti-cancer drug, which is quite interesting. It has been developed as an inhibitor of the EGF receptor-associated tyrosine kinase activity. And that should be followed up further, especially because it does not bind to the active site of the main protease, it binds to the allosteric site at the domain 3 dimerization site. So through this very high effort activity of the DAISY colleagues, there's more than 100 colleagues on the publication, on the paper, which has been submitted. The main merit was to map, in my eyes, was to map these allosteric sites. So now we know that we have other attachment points, not only the active site for the main protease. We also ourselves try to engage in drug repurposing, and we centered our activities on the alpha-ketoamides, which have been FDA approved about 10 years ago as inhibitors to block the hepatitis C virus, NS3-4A protease. And these are boceprivir and telaprevir. So these are alpha-ketoamides, and we co-crystallize boceprivir with the SARS coronavirus main protease. And you see the formula I guide you through from the right. You have the 2 keto groups, the alpha-ketoamide from the C terminus, so to speak, of the compound. Then in P1, you have a cyclobutyl methyl group rather than the cyclic gamma lactam. So this one cannot make hydrogen bonds, it's hydrophobic. Then in P2, you have a large entity, a bicyclic proline derivative. And we found out that this bicyclic proline derivative fits nicely into the S2 pocket of the main protease of coronavirus because of the plasticity, which I described before. So again, the glutamine 189 moves out of the way, this large group binds. But the cyclobutyl, it's also located to the S1 pocket, but as I said, it cannot make hydrogen bonds. So we create less of binding energy here. The Ki is, according to our measurements, by a factor of 4 higher than that of our compound 13B. The IC50 is above 5.8. So activity at the enzyme is too high. It's too low, excuse me. And in virus-infected cell culture, it is disappointing. So another approach of repurposing, which did not lead to much. We are now busy replacing this cyclobutyl group by the gamma lactam and create a hybrid which should be very active against the coronavirus main protease. But then, of course, we lose the advantage of drug repurposing, which is to have a drug which is already approved for other purposes, of which we know is not toxic, and we don't have to go through all the toxicity measurements, and we can have shortened clinical trials for fast approval. So far, drug repurposing was disappointing. Another approach is fragment screening. What you do in fragment screening is you work with libraries of small fragments, molecules, chemical entities with molecular weights less than 300 Dalton. And then you determine by X-ray crystallography, or you can also use NMR, SCD, NMR in particular, to locate the binding sites of these fragments on the target structure. And then if you have two adjacent fragments bound to adjacent pockets, you link them by chemical means. And that approach we follow together with the colleagues at BESSY, Manfred Weiss and Jan Wollenhaupt. And this slide was prepared by Jan for me. So what you do is you have a library, they call it F2X, of small molecules, fragments, and you screen your crystals of the target Mpro against these fragments. And then, of course, you get many empty structures where nothing is bound. You have to determine all these, but you get some where fragment is bound. And then they found some fragments bound to the target enzyme, and we are linking them right now. This shows the molecular weight distribution of their library. And it's shown in purple here. Also, the yellow one is another fragment library. So the molecular weights are actually around 200 Dalton, whereas the fragment library, not the fragment, the drug library, the repurposing library used by the DESY colleagues is indicated in blue. That has higher molecular weights above 300 up to 1000. So we call this project together with the BESSY colleagues the Stop Corona project. It's funded by the BMBF. So the project is administered by DESY PS. And I'm very grateful for this collaboration and this funding. So far, the colleagues at BESSY have found 3 fragments bound to the active site, 1 bound to the dimerization interface at that allosteric site I mentioned before. And they have already linked 2 fragments and created a non-covalent binder, which is linked, consisting of 2 fragments. And now we are busy attaching a covalent arm to attach it to the active site cysteine and increase the affinity to the main protease and turn it into a good inhibitor. Obviously, this will take time, and we have to test the inhibitor then for toxicity and for activity in infected cell culture. And we also have to do structure-activity relationships, so synthesize many derivatives. But we want to be ready for SARS-3 with this compound. We should not do the mistake again, like it was done after SARS-1, that everyone says, okay, the pandemic is over, we are no longer interested, let's forget this bad experience and do nothing for the future. We protect ourselves against so many probable or improbable events. Only think of military aggression. But we don't protect ourselves enough against the next outbreak. And this has to be changed. I hope people will learn this after the current pandemic. Even though with antiviral drug development, you cannot earn the rapid political success that you may hope to earn with vaccination. So let me summarize my talk. I have hopefully shown you that structural biology, X-ray crystallography, can act very rapidly. Within 3 weeks of the publication of the genomic sequence of the new virus, we had the crystal structure of the main protease. Thanks to the help of Manfred and his colleagues at BESSY, who gave us beam time generously. We used the structure to optimize our existing alpha ketoamides. If we had not worked on these inhibitors before the current outbreak, we would be far from having these compounds. We have shown that since the publication of our work, hundreds of groups, literally, in the world are using the main protease structure to design their own inhibitors, some of them with good success. I mentioned the Pfizer compound. Virtual screening for drug repurposing has been disappointing so far. The massive screening of existing drugs by BESSY has yielded at least one interesting compound, but the effort was huge, with 5,900 plus compounds co-crystallized with the main protease. I mean, attempted to co-crystallize. 37 of them did co-crystallize. They have found the allosteric binding sites. Repurposing HCV-protease inhibitors was only partly successful, but it pointed a way to hybrid molecules, which might be better drugs against the coronavirus main protease. And fragment screening is a big hope, which we have to come up with completely new ideas for non-peptidic inhibitors for SARS coronavirus 3. I like to acknowledge my co-workers. Chemical synthesis in the group was done by Daisong Lin. Protein purification and crystallography, Lin Zhang, Xin Yuan Sun. Enzyme kinetics, Lin Zhang, she's a key person in this project. Collaborators for chemical synthesis, we now collaborate with the Helmholtz-Zentrum, Mark Bröser's group in Braunschweig. Crystal structures, we collaborate with BESSY, Jan Wollenhaupt, Manfred. We also are part of the big DESY consortium under the leadership of Alke Mainz. Cell infection assays are performed by the group in Marburg, Stefan Becker by the group in Leuven, Johann Neitz. Pharmacokinetics by Katharina Rox. I thank you for your attention. I'm ready to take questions.
Well, thank you very much, Rolf, for this really interesting overview and lecture on the structural biology of drug discovery using the main protease. You should imagine the applause of roughly 360 people now. We had 160, 170 in the room, and a further 200 were following on the HZB YouTube channel, and this was on which it was broadcast live.
Oh, great. Thank you.
Thank you very much indeed. I have the first question from you, from Milton Stubbs in Halle.
Oh, Milton yes.
Asked, could you say something about the relative attractiveness of inhibiting the main protease versus, for instance, the papain-like protease and the and other enzymes?
Yeah, the papain-like protease, in my view, is a more difficult target. I'm also working on that. It is more difficult because it leaves after glycine. That means that the S1 pocket and the S2 pocket are very small. And it's difficult to find non-peptidic entities which can bind here. It's also difficult to make covalent inhibitors. However, the S3 and S4 pockets, they are useful targets for virtual screening, actually against the SARS-1 papain-like protease. An inhibitor has been found by virtual screening. It's called GRL617. It was found by the Mezika group in 2008, has been optimized by medicinal chemistry. And this is a non-peptidic, non-covalent binder, quite interesting, and also active against SARS-2. So for virtual screening, these two pockets are attractive, and it should be done. Another complication is that the papain-like protease has a blocking loop, BL2. And this loop has different conformations and different constitution in different enzymes, and from different coronaviruses. So it is longer in MERS coronavirus than it is in SARS coronavirus, and it partly blocks the active site. So it will be difficult to come up with pan-coronavirus inhibitors, which can not only inhibit SARS-2, papain-like protease, but also MERS and other papain-like proteases. Milton also asked for other enzymes.
Yeah.
Of course, the other prime candidate is the RNA-dependent RNA polymerase. We have good cryo-electron microscopy structures now in sufficient resolution to do drug design of this enzyme, NSP12, in complex with its cofactors NSP7 and 8. And for instance, remdesivir has been shown to bind to that site, and the mechanism has been revealed by groups in China and also by Patrick Kramer in Göttingen. And this is attractive, but so far we are bound to nucleoside and nucleotide analogs. Another attractive target would be NSP13, the helicase, and some work on this has been done at the diamond synchrotron. And also NSP14, the exonuclease, is attractive because with this large genome, 30 kilobases, the coronavirus needs proofreading. We all learned in our virology lectures years ago, at least the elder ones among us, that RNA viruses have no proofreading. But the coronaviruses do have proofreading, because otherwise they would not be able to maintain the integrity of this huge RNA genome. So they have to cleave out mismatches. And this is done by the exonuclease NSP14. So if you inhibit the exonuclease, the proofreading process, then you can steer the virus during its evolution to an error catastrophe. And you may combine this with an RDRP inhibitor, and then you may achieve good results. But we are a little bit far from this, so nobody yet has described a good NSP14 inhibitor. So this huge genome offers many points of potential interference with the viral life cycle. That is a good thing. But the bad thing is that the life cycle is much more complicated than with many other RNA viruses.
Okay, thank you. There's another question by an anonymous participant about boceprivir, which you mentioned, and you have started to modify it. But this question is there a potential to repurpose boceprivir and other drugs to really administer it against COVID-19?
Well, in our hands, boceprivir had an IC50 of 5.8 micromolar. That is too high. That's the isolated enzyme. In virus-infected Calo3 cells, it simply did nothing. There are some reports from China that it maybe have some slight activity. We cannot reproduce that.
Okay, thank you. And then a more general question, also by an anonymous participant. Is a simple N-methylation of an amide enough to hide the amide bond in favor of desirable pharmacokinetics?
You can think of that to amidate to attach a methyl group to the amides, but you need the nitrogens for hydrogen bonding. That is a problem. So then you lose this hydrogen bonding. In the case of the P2 nitrogen, which is the one which we hide in the pyridone, then you don't need the hydrogen bonding. There is hydrogen bonding if you don't modify it to glutamine 189, the famous one which I mentioned before, but you can still get good activity if you destroy that hydrogen bond. So you may also be able to selectively methylate this nitrogen and get a good binder. But I'm not sure there is a space for the methyl group. With the pyridone ring, the advantage is that you don't need extra space because you have this cyclic structure inside the binding site, within the binding site for the inhibitor. So it's worth maybe trying for that nitrogen, but you have to be selective. You cannot methylate the P1 nitrogen.
All right. Milton says, thank you for the exhaustive answering of his question and sends schöne Grüße from Halle.
Danke, schöne Grüße zurück.
And at this stage, I would like to close the discussion and thank you very much, Rolf, again for this nice lecture. Before everybody leaves, I was supposed to tell you to please move back to the lobby because there is some final words by the organizers before you all go home. So thank you very much again, Rolf, for this lecture, and have a nice evening.
Manfred. Thank you, everybody. Yeah, thank you, everybody, for listening.
Thank you also.
Sure.
Just an organizational note, it might take a few seconds, a minute or two until we have the live stream going in the lobby. So if you don't see anything yet, just be patient. And with that, I'll send you off into the evening and have a great evening.